All right, this is nucleic acids part 2. You may think, oh, well, this is still part 1, but in fact what we're doing is looping back. So we're taking a look now at these information molecules that we call DNA and RNA and looking at the differences between RNA, which has a ribose sugar, which is, remember, it's a pentose, so it's a 5-carbon sugar. That OSE ending, remember, also tells you that it's a sugar because OSE represents sugars. It has one of these 4 bases. And when I say one of these 4, it's going to be made up of a combination of these, but it's not going to pair with another one because it's single-stranded. And then DNA has a different type of sugar called deoxyribose. It has these 4 nitrogen bases. Notice that the only real difference is that uracil is found in RNA and the base thymine is found in DNA. And DNA is double-stranded. So this is the typical way we see these nucleic acids. More often than not, we're going to see it as represented by this double-stranded, this double helix. The other thing to note is that when we're talking here about DNA, what we see is that there's always the same pairing. So if this was one strand on this side, and remember, it's double-stranded, so there'd be another strand of DNA on this side. Wherever you have thymine, it's going to bond to adenine. No matter which side it's on, it's always going to be A's to T's or T's to A's with a double hydrogen bond. Cytosine is always going to bond to guanine, and there will be a triple or hydrogen bond. So notice down here, what it's trying to show you is cytosine to guanine. These are, there's 3 bonds between different parts of these nitrogen base pairs. See where the nitrogen is. On this one, you see a couple of nitrogens up here. Here's another nitrogen over here. So just know that these colored little geometric shapes, whether they're single rings or whether they're a fused double ring, these are the nitrogen bases. And then here, this is the sugar. So what we're seeing here, we aren't seeing the whole picture, right? We're just seeing now this part here without the phosphate group, which would be over here. So this officially, if I'm only looking at this, I can call it a nucleoside. When we put the phosphate group on there, then we're going to call this that unit. That unit is the monomer of DNA, so that's a nucleotide. So this is complementary base pairing. Now why this is so important is because those pairs and their sequence up and down a strand of DNA builds our chromosomes, right, in our cells. This is the information that makes you you. All of these they're going to alternate. They're going to be different codes in a sense for different kinds of proteins. So DNA is really a code for the myriad of different types of proteins that you have that make up your body, make up body chemicals, all of that. Now the hydrogen bonds are pretty strong in DNA and in RNA, in part because when DNA forms this double helix, these hydrogen bonds aren't really exposed to the water or the liquid environment that's around it. So they're kind of protected from water. So they're pretty strong, but they can still be broken. And so if need be, which is what the cell does when it wants to expose some part of the DNA to create a particular protein, in other words, it's going to find a gene, then it needs to break those base pairs so it can make a copy of one side of that strand. Now RNA, the single-stranded structure, is really found in 3 different types. We have RNA that is the one that makes the copy of a part of a DNA molecule. And that's going to be messenger RNA. So messenger RNA is going to be able to make a copy, and it's going to move that information out of the cell, out of the cell's nucleus, to the place in the cell that makes the proteins. But here, what we're talking about is that mostly we call RNA is a single-stranded structure, but it can fold, and it can fold. So here's its basic structure. Here you can see would be a nucleotide because it's a phosphate group. Here it would be ribose sugar. And then on this one, you're going to see there's the nitrogen base. But what you see here is that it's single-stranded, and we're running from what's called, remember, the 5' end up to the 3' end. And we're basing that sort of orientation of this RNA molecule. When we say 5', look right here and see that represents at the bottom of the strand. Here's the carbon number 5, which is this the bottom. But then at the very top, we can see up here we have the 3', and it's sitting on that third carbon of this ribose sugar, where this is the orientation is the 5th carbon. So that's kind of what we're talking about here, although we'll elaborate more later. But here's how it can fold. So if you took this molecule and made it a single strand, because that's what it would start as, but it has the capability of folding into various shapes, and some of that folding would see if you could kind of magnify this a bit. I don't know if I can. Nope, I guess I can't. But what you'd see is that there's some complementary base pairing that's occurring along certain parts of this long strand that sort of hold it together. And that's what we see here. DNA, though, is a polynucleotide, which just means there's a lot of nucleic acids or monomer units that are all joined together, but it has 2 of them, and they join together by complementary base pairs, and then they're going to form that double helix ladder. And what you see is that the nitrogen bases are the ones in the middle because they're bonding either by 2 hydrogen bonds or 3, and then the outside, if you kind of look at it as if it's a ladder, would be the sugar and the phosphate groups. So they'd be the outside of the rungs. Sorry, not the rungs. They'd be the sides of the ladder where the hydrogen bonds would be the rungs. So here it is. This may be helpful. So here's the outside on this. So if it's a ladder, remember it's going to twist actually. So we're seeing it straight. But here's the nitrogen bases held together by hydrogen bonds. Here's the outside of this molecule with the phosphates and the sugars that are sort of on this outside piece, and also over here. Notice that it has its orientation is that one side is going from 5' to 3, just meaning here's the bottom of it here is the 5th carbon, and at the top you see that last one is we're looking at the 3rd carbon here, but then the other side is the flip side of that. So then we have a 5' to 3' end. Now you might say, well, why? I hope you do, because it is a little bit curious, isn't it? So there are a few things about this molecule that are really interesting. And the scientists by the name of Watson and Crick, and we'll give some credit we should give a lot of credit to a woman by the name of Rosalind Franklin, who really gave them the tools that they needed to figure out this structure. And they got the Nobel Prize initially for that. But what they figured out is, look, the evenness of this ladder is partially because of the way these are arranged. So we have purines and pyrimidines that are always alternating. So these A's, T's, C's, and G's, you always have one that has 2 fused rings, one that is a single, all the way up and down. So what that does, it does a number of things, but one thing it does is it keeps the width of this molecule even. So no matter what letters we have, we know that a C is always going to, the nitrogen base that has cytosine will always bond by those 3 hydrogen bonds to the one that's a guanine. And so whether it's on the right or the left, it doesn't matter. It's always going to bond there. So you're always going to have a 2-ring structure and a single ring structure. There's a 2-ring structure and there's a single ring structure. And up here, you can see that this one is in reverse of this. But the width of this molecule is the same. The other thing we'll talk about a little bit later when we look at how proteins are actually made is why there's some significance to having this triple hydrogen bond here, and where we might find that in the DNA molecule, and the 2 hydrogen bonds, so the 3 versus 2. So we see the pyrimidine here, purine here, et voila. So a little bit of a repetition, but it takes a little bit of time to kind of look at this molecule, really think about why is this so interesting and important, and that it kind of blows you away if you think about the fact that we're really talking about an alphabet of only 4 letters, and that's A, T, C, and G, and that those 4 letters, in whatever combination they have up and down these very, very long DNA molecules, really spell out the code for a particular protein that your cell would need. So we'll talk more about that in a few more episodes. Here's the ladder, the double helix. You can see that one side has that 3' and the other has the 5, and it's twisted. You can see in this case that it's an evenly spaced structure, so that otherwise you might end up with something that's kind of like too wide here and then narrow here. Instead, it's very even because of that arrangement. And then finally, really to the crux of it is, what is DNA? And what is DNA? It is the information. That sequence of base pairs is a code for a protein. And what DNA has to be able to do, it has to be able to copy that whole, all of the DNA in that organism have to be copied in order to send that information on to the new cells. So when new cells are born, we've got to be sure that they have all of that information for the next generation of cells. And then they are able to cause certain genes to get expressed, meaning it's going to be translated into a protein. So DNA builds RNA, I should say this differently. RNA is built from a certain part of a strand of DNA. Some of that RNA is able to go out into out of the nucleus of a cell, and we're going to see that there are 2 other types, actually 3, of RNA that exist outside the nucleus that help in the process of making a protein or a polypeptide. When you see how things are lined up, it's kind of nice when you see this. If I gave you a strand that said, here's the top, and we're going from 5 to 3 on this top one, and the first nitrogen base is thymine, second is cytosine, third is adenine, guanine, cytosine, adenine, and I'd ask you, tell me what the complementary side looks like, then you would just simply go the opposite way from 5 to 3 this direction, but then be sure that across from each other are the ones that actually bond together. Always A's to T's, always C's to G's. Now, I'm going to have to stop here, but I think we've gotten to a good spot. Here's the RNA, and RNA can get transcribed, and we'll see how that works later. But notice what happens there. So when a strand of RNA gets converted into a different strand, and we'll explain later why, instead of having a T on this side, we see the U, and then everything else is the same. So wherever you see, so T uracil is going to replace this little strand that you see here. This would be actually the DNA strand that was being converted into RNA. So I think I confused you there, but I'll clarify in class. And the whole set of DNA in an organism is called its genome. So there you go.